Hello everyone, I'll introduce the paper submitted to iMetaMed. The title is, A Cell-Penetrating Scorpion Venom Peptide Disrupts the HSP90β-PPARα Interaction to Ameliorate Metabolic Dysfunction-Associated Steatotic Liver Disease. Highlights of this study: 1. Discovery of a novel cell-penetrating scorpion venom peptide BmK Tx-2. Improves MASLD. 2. HSP90β acts as a repressor of PPARα activity. 3. Dissect the precise molecular interactions between HSP90β and PPARα. 4. Dissolution of HSP90β-PPARα interaction is a potential therapeutic strategy for MASLD. 1. BmK Tx-2, isolated from the venom of the BmK scorpion, attenuates MASLD and MASH. We found that Buthus martensi Karsh scorpion venom markedly alleviated hepatic lipid accumulation, improved liver function, and enhanced insulin sensitivity in high-fat diet-induced mouse models of MASLD through activity-guided fractionation. A scorpion venom-derived peptide composed of 66 amino acids, termed BmK Tx-2, was identified. This peptide effectively reduced lipid accumulation in multiple hepatocyte models and in vivo mouse models without detectable cytotoxicity. Notably, in two MASH models, BmK Tx-2 significantly ameliorated steatosis, inflammation, and fibrosis, with overall therapeutic efficacy comparable to that of the positive control drug resmetirom. 2. BmK Tx-2 is a cell-penetrating peptide that can be internalized by hepatocytes. In vivo tracing experiments showed that BmK Tx-2 predominantly accumulated in the liver after administration, indicating strong hepatic tropism. Cellular studies demonstrated that BmK Tx-2 actively internalized by hepatocytes in an energy-dependent manner, primarily through macropinocytosis mediated by the RAS-PAC1 signaling axis. Furthermore, BmK Tx-2 efficiently escaped from endosomes following cellular uptake, providing the mechanistic basis for its intracellular target engagement and biological activity. 3. Activation of hepatic PPARα is required for BmK Tx-2 to attenuate MASH. Transcriptomic analyses of hepatocytes and liver tissues revealed that BmK Tx-2 treatment profoundly reshapes metabolic and inflammatory gene expression programs in MASH, with the PAR signaling pathway emerging as the most significantly affected core pathway. Mechanistically, BmK Tx-2 selectively enhanced hepatic PPARα expression and transcriptional activity, upregulated multiple genes and proteins involved in fatty acid beta-oxidation, and markedly increased fatty acid oxidation rates, thereby alleviating MASH pathology. Loss of function studies further demonstrated that knockdown of PPARα in hepatocytes or whole-body deletion of PPARα in mice completely abolished the therapeutic effects of BmK Tx-2, establishing that its anti-MASH efficacy is strictly dependent on hepatic PPARα activation. 4. BmK Tx-2-mediated PPARα activation is dependent on HSP90β. Proteomic target identification and functional validation revealed that BmK Tx-2 does not act as a direct ligand of PPARα, but instead specifically binds to the molecular chaperone HSP90β. Knockdown of HSP90β in hepatocytes markedly attenuated BmK Tx-2-induced activation of the PPARα signaling pathway and its lipid-lowering effects, indicating that HSP90β is required for BmK Tx-2-mediated PPARα activation. In vivo, hepatic overexpression HSP90β completely abolished the protective effects of BmK Tx-2 against MASLD, demonstrating that the therapeutic efficacy of BmK Tx-2 is critically dependent on the HSP90β-PPARα axis. 5. BmK Tx-2 disrupts the HSP90β-PPARα interaction, leading to PPARα nuclear translocation. Analyses of clinical samples and functional experiments consistently demonstrated that HSP90β is markedly upregulated during MASLD, MASH progression, and directly binds to PPARα, restricting its nuclear translocation and transcriptional activity and impairing hepatic lipid metabolism. BmK Tx-2 does not alter HSP90β protein abundance. Instead, it specifically binds to HSP90β and disrupts the HSP90β-PPARα interaction, release PPARα from cytoplasmic sequestration and promoting its nuclear accumulation. Multiple complementary approaches, including colocalization analysis, co-immunoprecipitation, pull-down assays, and biolayer interferometry, collectively confirmed that BmK Tx-2 enhances PAR-alpha nuclear translation and transcriptional activity by disrupting the HSP90β-PAR-alpha interaction, thereby driving fatty acid oxidation and exerting therapeutic effects against MASLD. 6. V55 of BmK Tx-2 binds to K349, R405, K406, and D444 on HSP90β to disrupt the HSP90β-PAR-alpha complex through molecular docking, site-directed mutagenesis. Multiple biochemical and functional assays, we precisely defined the key interaction interfaces between BmK Tx-2 and HSP90β. BmK Tx-2 competitively binds to the middle domain of HSP90β, overlapping with the PAR-alpha binding interface, and primarily relying on four critical residus K349, R405, K406, and D444 on HSP90β. Mutation of these residues markedly weakened the HSP90β-PAR-alpha interaction and abolished the disruptive effect of BmK Tx-2. On the BmK Tx-2 side, V55 was identified as the key residue mediating HSP90β binding and biological activity, as the V55 mutation almost completely eliminated its ability to disrupt the HSP90β-PAR-alpha interaction, activate PAR-alpha signaling, and promote fatty oxidation. Together, these findings elucidate the structural and functional basis by which BmK Tx-2 binds HSP90β at defined residues to competitively relieve HSP90β-mediated repression of PAR-alpha. This study identified a natural cell-penetrating peptide, BmK Tx-2, derived from Buthus martensi karsh scorpion venom, which markedly ameliorated lipid accumulation, inflammation, and fibrosis in multiple mouse models of MASLD. Mechanistically, BmK Tx-2 specifically binds to HSP90β via its V55 residue, disrupts the HSP90β-PAR-alpha interaction, promotes PAR-alpha nuclear translocation, and subsequently activates fatty acid beta oxidation. These findings reveal HSP90β as an endogenous negative regulator of PAR-alpha-driven lipid metabolism, propose targeting the HSP90β-PAR-alpha interaction as a novel therapeutic strategy for MASLD.